Okay, so I'm Maria Bernabeu. So I started in Barcelona here at UPF. I did my bachelor's and master's here, and then moved to Seattle for a postdoc, and then came back to EMBL to start my own group, which is focusing on two main topics, malaria pathogenesis, and we're using vascular engineering models to study this pathogenesis. So we've been hearing about COVID a lot, like every day in the last year, but malaria has been with us for billions of years, and it's still a huge public health problem. It causes every year about 200 million cases. And it has an extremely complex life cycle. So it's transmitted by a mosquito that injects these infective forms, porozoites, that travel to the liver, where they have these rounds of asexual multiplications. After they multiply, they go back to the blood where they will infect red blood cells in a 48-hour cycle. And then eventually, some of these asexual parasites will commit into asexual stages known as gametocytes, which will be taken by the mosquito. And then in the mosquito, we have this sexual replication of the malaria parasites of the gametes. And of course, something like this, so complex, can only be done by a protozoan parasite. There are more than 100 species, malaria species that infect birds, reptiles, and mammals. And there are 5 of them that infect humans. Plasmodium falciparum and Plasmodium vivax are the most common ones, the most widely distributed. And one of the things that attracted me to malaria first is the fascinating cell biology of the malaria parasites. So it's an eukaryotic cell. Of course, it has nuclei, mitochondria, but also has some specific organelles. So the picoplast, this is a chloroplast-like organelle, non-photosynthetic. We also have rhabdoids and micronyms that are organelles for secretion of proteins during invasion. If we go to blood stages, and I will talk about this later, we have an amazing distribution of a modification of red blood cells. But my lab focused on this interaction between malaria parasites and blood vessels. And I really wanted to focus on blood vessels because malaria parasite takes blood vessels to go through the human body and complete its cycles. So first we have porozoites that are injected in the skin and then go right away to blood vessels. Then in blood vessels, they will infect hepatocytes, but after multiplication, they will come back into these blood vessels. Then at the end of the cycle, of course, it's also very important. So these committed sexual parasites will go to bone marrow and then go from the bone marrow, from the blood vessel to the bone marrow, then again from the bone marrow to the blood vessel, and we'll end up this malaria cycle by going from the blood vessels into the skin and to the proboscis of the mosquito. But the main focus of my lab is not these malaria stages, it's the blood stages, because besides infecting red blood cells, malaria-infected parasite red blood cells accumulate in the microvasculature to escape spleen clearance. So there's accumulation in all body, but this accumulation is very big in certain organs. So there's big accumulation in the gut microvasculature, and this causes metabolic acidosis, so decrease in the body pH. There's big accumulation in the placenta in pregnant women, and this causes miscarriages and big burdens on maternal mortality. And there's also, the main focus of my group is parasite accumulation in the brain microvasculature, which causes cerebral malaria. And in cerebral malaria, so you can see here, there's this big accumulation of parasites in the brain microvasculature, and there's here this microvessel full of parasites, which is this black dots here. And this is very different from a normal brain blood vessel, and this happens in all the human brain, leading to this massive brain swelling. So you can see here an MRI, brain of an uncomplicated malaria patient compared to a brain of a cerebral malaria patient. There's this massive brain swelling caused by malaria parasite accumulation in the brain, which causes a compression of the brain stem here. And these kids here stop breathing because of this massive brain swelling and compression of the respiratory center in the brain. So this causes every year about 500,000 of deaths, mostly in children under 5 years old. And when we study cerebral malaria, we don't have access to the brain of the patients. So malaria biologists have been using different models to try to understand these complications. So during my PhD and my postdoc, I used thousands of times. I grew cells in monolayers and put Plasmodium falciparum on top. So endothelial cells grow on a plastic dish, and then we put parasite on top. And this is great. It's very easy to learn, and it's kind of high throughput, but it's very microfluidics with 3D culture and organoids to overcome these limitations of the lack of models in cerebral malaria. So this is how our model looks like. So we have two collagen pieces, one flat collagen piece at the bottom, and then one collagen piece at the top that has an imprinted microfluidic network. So we can choose the design of the microfluidic network that we want. And when we put these two pieces together, we end up having this collagen hydrogel crossed by open tubular structures. We can seed endothelial cells in these tubes. And we can also seed other cell types of the surrounding tissue in the collagen matrix. So I started this during my postdoc in Seattle in the lab of Joe Smith and Ying Zheng, in a collaboration with Ying Zheng, who developed all this technology. And this is how our 3D microvessels look like. This is a brain microvessel model with primary brain endothelial cells. And you can see how the cells love to grow on the collagen and have beautiful adherent junctions. When we seed the endothelial cells within our system, we end up having this beautiful lumen that can be perfused with red blood cells. We can disassemble these devices and bring it to the electron microscopy. And we can see the beautiful cell biology going on in these cells. We have caveoline polarization on the abluminal side. We also have formation of very complex junctions, and I will go back to this later. And so this is how our model looks like. This is a grid network comprised by 100 micrometer channels, and you can see here in this 3D reconstruction how we have these open luminal structures. And the main advantage of our method compared to other 3D vascular engineered models is that we can control very, very well the microfluidic properties. So one of our favorite geometries is this grid here that recapitulates a wide range of flow velocity and wall shear stress. So we can model physiological blood flow and pathological blood flow within the same device. We can also model flow acceleration and deceleration here. And this is an example of this grid model perfused with Plasmodium infected red blood cells. You can see this is the same device imaged at 2 different regions, and you can clearly see the difference in the flow velocity, how red blood cells flow through it. This is a moderate wall shear stress, moderate velocity region. This is a parasite has a strategy, so it has a repertoire of different 60-bar genes that have antigenic variation and clonal expression. So only one PFMP1 is expressed at the time of each infected red blood cell. And these proteins mediate cytoadhesion to endothelial receptors. They mediate cytoadhesion to some of them, of these PFMP1s bind to both endothelial protein C, EPCR, and ICAM1. There's another subtype of these 60 different genes that only binds to EPCR. And then there's other cell types that bind to other parasite types that bind to C36 and ICAM1. So I'm going to focus on these 3 today, but you can also know that there's other PFMP1s that mediate rosetting. So it clusters uninfected red blood cells around the parasite to hide them from the immune system and facilitate invasion. So in our study, we use these clonal lines expressing a single PFMP1, each of these binding to multiple receptors. And I just want you to know that the 3 lines that we use are highly expressed in cerebral malaria patients with brain swelling. So we've been using these cell lines for a long time in the lab for years and years. And we always found that when we do cytoadhesion experiments on 2 conditions, this strain here, variant 10, who expresses an EPCR binder, presents super high binding to endothelial cells. But these other cell lines that are highly expressed in malaria patients present very low sequestration rates to endothelial cells. So we perfused these three cell lines through our system. And this is what we got. So in this graph, I'm plotting the shear stress here at the bottom. So we have here regions near the inlet with high shear and regions in the corner with low shear. And as you can see here, all our parasite strains present shear stress depending bindings. So their favorite site of sequestration is regions of low shear stress. And very surprisingly for us, all our cell lines perform equally well when they cytoadhere in 3D at these physiological flow levels. And this one, even one of them presented significantly higher adhesion in these shear stress depending levels. So our model better mimics the flow in the microvessels, and parasites cytoadhere much better into them. And then these studies that I've shown you here were done kind of reproduce what happens in the early infection when we have a resting endothelium. But this is quite different what we have in severe patients where we have a super activated endothelium. And this activation causes ICAM1 upregulation at the surface of infected red blood cells. And we also have a loss of EPCR through shedding in these endothelial cells. So we mimic these conditions by stimulating our 3D microvessels with TNF-alpha overnight. And we found that our parasites present a very different behavior. So it was believed that parasites activate the endothelium to increase their binding and promote parasite survival. And this is what we found with this strain. You can see a massive upregulation of binding here in red when we stimulate with TNF-alpha. But with the other two strains that we use differently to us believe in the malaria field, we found that they do not present this upregulation of binding. So there's this strain here that presented a slight downregulation. And that's this strain here that presented no changes in cytodhesion at all. And this is due to this downregulation of EPCR at the surface of endothelial cells. And last, we wanted to prove that this sequestration was mediated by these endothelial receptors. So before perfusing our parasites, we incubated our vessels with inhibitory antibodies against EPCR and ICAM1 for 30 minutes, and then we perfused the parasites. And with all the parasite lines, you can see in red the inhibitory conditions. We found downregulation, significant downregulation in the binding, meaning that these two receptors are mostly mediating parasite sequestration and binding to endothelial cells. So all these studies that I've shown you here have been done with these 100 micrometer vessels, which mimic arterioles and postcapillary venules. But our lab is mostly interested in also studying these capillary interactions with parasites with capillaries, which are way, way smaller. And I've mentioned that we can use different microfluidic networks, but we cannot go down to 50 micrometers with this fabrication method. So we improve our fabrication method to get smaller vessel sizes, and we use a different methodology. So first, we created 2 big channels, parallel channels with soft lithography, as I've shown you before, and we seed endothelial cells and let them grow for a couple of days. Then we bring these devices to the 2-photon microscope and we photoablate small capillaries size vessels here in the collagen. We bring these vessels back to the incubator, and after a few days in culture, we have migration of endothelial cells into these narrow regions, forming these capillary size vessels in the central region. And we found that this hourglass shape was key and essential to have this migration of endothelial cells to these super narrow regions. So we have vessels that range from 40 micrometers to 5, 10 micrometers in this narrow region. We can also bring these samples to the electron microscopy. You can see these beautiful capillaries that range from 40 to 20 to 10 micrometers. And it's quite fantastic to see how on this 5 micrometer region, we have a single endothelial cell wrap around and forming this capillary. And most important, we can perfuse these capillaries with blood at full hematocrit, physiological hematocrit, 40%. And this is one of my favorite videos here. You can see this fascinating blood transition from the precapillary region to the capillary region. We only have 1, 2, or 3 red blood cells flowing at a time to this post-capillary region here. And this system here is ideal to understand how microvascular in small capillaries occurs. And we wanted to study that because there's been big debate in the field of how this microvascular obstruction occurs. First, there's this biological hypothesis. So we have sequestration and active ligand binding to a receptor in endothelial cells. I've explained a little bit at the beginning that the malaria parasite also remodels completely the red blood cell membrane here. and creating these knobby structures that cross-link the membrane cytoskeleton, increasing the rigidity of the red blood cells. And last, we can have a purely mechanical hypothesis. So we have a red blood cell that is very extremely flexible with a parasite inside, which may cause mechanical obstruction. And to understand that, we did a very simple but very conclusive experiment. So we perfuse uninfected red blood cells and 3 different versions of infected red blood cells. So one is the wild-type parasite. It has the ligand PFMP1 on the top, and it has also knobs. We also trypsinize PFMP1. And by doing that, what we did is to shave and remove the ligand from the surface, but still maintaining the knobs. And finally, we use a knobless parasite line expressing a very similar PFMP1. So we have PFMP1, but we don't have knobs. And we perfuse these parasites. And when we perfuse uninfected blood cells, as I've shown you earlier, there's no accumulation at all because these red blood cells are extremely flexible. But then when we perfuse these 3 cell lines, you can see that we have very different behavior. We have very different accumulation patterns within the microvessel. So we can calculate everything at the end. And what we found is that this wild-type parasite with PFMP1s and knobs accumulates in these high shear stress regions of the capillaries. When we remove the ligand from the surface, what we found is that there's no sequestration at all. So what we found is that the sequestration levels were not significantly different from uninfected blood cells, meaning that the ligand and this active binding to endothelium is key for parasite sequestration. And when we perfuse this knobless line that has PFMP1, so it has a less rigid cytoskeleton, what we found is that parasites still sequester, but they only do it in this region here of the capillary when there's flow deceleration and there's more red blood cell tumbling. So what we conclude from this is that PFMP1, this ligand, is essential for sequestration, and the presence of knobs in the surface and cross-linking of the cytoskeleton increases the affinity of the parasites to regions with higher shear stress. So just to conclude this first part of the talk, I can I've shown you that we can use these 3D vessels, this 3D engineered system to study malaria pathogenesis. We found that these systems better reproduce the flow microvasculature, and parasites present better binding on it. And we found that contrary to what it was believed, not all cell lines present upregulation of binding after endothelial activation. And we also found that PFMP1s were key for this interaction. And this is what I did mostly during my postdoc, but then a year ago, I moved to Barcelona, and we want to go one step forward. And we would like to understand what are the pathogenic mechanisms of this disease and how we can go from this massive sequestration that we see in our capillary systems to this sequestration that happens in patients. And our models, the models that I've shown you before, are endothelial only and are quite leaky. And this is because they don't have, it's not a proper blood-brain barrier, which is composed not only by endothelial cells, but also by pericytes and astrocytes. And this coordination and crosstalk between these 3 cell types increases endothelial cell permeability, improves the junctions, and improves the transport of molecules in platelets, can also disrupt this blood-brain barrier in malaria. So I would like to finish. So most of the work I've shown you today was done during my postdoc in Seattle. So I would like to thank Joe and Ying for this great mentoring. And this is our COVID-friendly picture of the lab. So we've gone through a 2-people lab into a 6-people lab in a very brief, short period of time. And we've been very fortunate to be awarded with an ERC starting grant recently. So progress in the lab will start advancing very fast soon. And I would like to thank with this modification of the EMBL logo that Jim Sharp did. So this is our EMBL logo transforming in these 3D microvessels with a red blood cell here. And you can see red blood cells flowing and starting sequestering here. And I'll be happy to answer your questions.